Well, good morning, ladies and gentlemen. Obviously, my name is Paul Boyd, not Boyled. But I want to thank you all for joining me, and I'm grateful for the opportunity to address you all today. The topic of my presentation, you can read it. So, the yin and yang is a familiar symbol that we may have all come across at one point or another. And for those who are not familiar with this symbol, let me provide you with a brief meaning. The yin yang symbol is captivating because it represents the idea of being in Taoist belief. Keep that in mind. Being refers to our conscious experience as living beings beyond just what we perceive as real. According to Taoist philosophy, being has two fundamental aspects: order and chaos. This makes sense because in every situation, there are things we can predict and understand, as well as unexpected elements that can surprise us. Another fascinating element about this symbol is the black side with a small white dot and the white side with a tiny black dot. And let me fix this real quick. There we go. This symbolizes that chaos can change into order, leading to significant transformations. Conversely, even within orderly and predictable situations, sudden events can propel individuals into chaos. Therefore, despite appearing to be opposing forces, they engage in a perpetual and dynamic interplay. In the yin yang symbol, the two halves complement each other, forming a harmonious whole. In parallel, RNA binding proteins have a specific regions or domains that complementarily bind to RNA molecules. These interactions are crucial for the proper functioning of RNA binding proteins, as they enable the proteins to recognize and bind to specific RNA sequences or structures. Moreover, RNA binding proteins play a crucial role in balancing different RNA levels in the cell, promoting stability or degradation based on their function, maintaining cellular homeostasis, and proper gene expression. This analogy highlights the significance of complementary interactions for the proper functioning of RNA binding proteins. Additionally, RNA binding proteins play a multifaceted role in regulating intrinsically disordered region proteins. Their interactions with IDPs can impact RNA metabolism, protein function, and cellular processes, making them key players in the dynamic and intricate network of cellular regulation. In the realm of IDPs, there exists a dynamic equilibrium between ordered and disordered states, akin to the fluid interaction of the yin and yang. IDPs continuously oscillate between structured regions and disordered segments, folding into defined structures during interactions or adapting to cellular tasks through conformational changes, exemplifying inherent dynamic balance. Unlike structured proteins with well-defined functions, IDPs serve as regulatory elements, interacting with structured proteins as flexible and adaptable partners, much like yin and yin complements yang's stable nature. Despite their ambiguous functionality, IDPs play vital roles in cell signaling, transcriptional regulation, and other processes. IDPs form intricate cellular interactions, engaging in multiple protein-protein interactions and participating in complex regulatory networks. IDPs influence various cellular pathways, much like the interconnected halves of the yin and yang symbol. Moreover, IDPs are crucial drivers and regulators of phase separation in cells. Their ability to undergo weak multivalent interactions and form dynamic assemblies allows them to influence the formation and function of membraneless organelles, impacting the cellular processes. In liquid-liquid phase separation, biomolecules segregate into two distinct liquid phases, mirroring the symbol's two halves. These separated liquid phases exhibit different properties and functions, creating a balanced system. LLPS involves a dynamic balance with biomolecules continuously exchanging between the phases based on cellular conditions, reflecting the fluidity and balance of the symbol. These phase-separated biomolecules can form membraneless organelles and structures, fostering specific cellular processes through interconnected biomolecular networks, much like the symbol's interwoven halves. In the cell, LLPS brings functional balance by organizing and compartmentalizing cellular components, enabling efficient regulation and coordination of biochemical reactions. Moreover, LLPS sensitivity to environmental factors like temperature, pH, and biomolecular concentrations reflect its adaptive nature, paralleling the symbol's responsiveness to change in environment. Lastly, phase separation is a fundamental process that underlies the formation and regulation of biomolecular condensates. Just as the symbol's halves complement each other, biomolecular condensates form through complementary molecular interactions, bringing together diverse biomolecules into organized liquid phases. These condensates constantly exchange biomolecules with the cellular environment, and the equilibrium between the two phases is crucial for their function and stability. Furthermore, biomolecular condensates, exhibit sensitivity to cellular cues and environmental changes, rapidly assembling and disassembling in response to their signals. This adaptability mirrors the yin yang symbol's adaptive nature, enabling cells to respond and adjust to different conditions effectively. However, abnormal biomolecular condensates can have far reaching effects on cellular processes and functions, contributing to the development and progression of various diseases. The yin yang symbol's complementary halves find a parallel in the functional duality of intrinsically disordered regions within RNA binding proteins. Mutations in IDRs can upset protein function, leading to more structured conformations that reduce RNA interactions or enhance disorder, affecting binding affinity. In RNA binding proteins, IDRs work harmoniously with structured domains and precisely, regulate RNA interactions. Mutations can disrupt this harmony, causing dysregulated RNA binding, impaired cellular processes, and disturbances in cellular homeostasis. The effect of mutations in IDRs depend on their context. Consequences may vary based on the specific RNA binding proteins, RNA targets, and cellular environment, emphasizing the importance of considering the broader context of mutation effects. Specifically, mutations in the RNA binding regions of the protein can impact its interactions with RNA sequences and structures, affecting phase separation behavior and influencing the composition, stability, and functionality of biomolecular condensates. These mutations have dynamic effects on disease, pathogenesis, with disease phenotypes potentially manifesting differently over time or in response to cellular conditions, making disease progression unpredictable and complex. Proper phase separation is vital for cellular organization and function. Mutations that impair normal LLPS can lead to dysregulated cellular processes, contributing to disease onset or progression. For example, the yin yang symbol's complementary halves find a parallel in the structure and function of TDP43's low complexity domain. Mutations in the low complexity domain disrupt the equilibrium between structured and disordered regions, affecting TDP43's normal LLPS, interactions with other cellular components, and cellular behavior. This imbalance contributes to the pathogenesis of neurodegenerative diseases, where abnormal TDP43 collection is a common feature. The yin yang symbol's balance between opposites reflects the functional balance required for normal TDP43 function. Mutation in the low complexity domain can disrupt this balance, leading to aberrant phase separation behavior, pathological aggregate formation, and impaired RNA regulation. These disruptions contribute to the neurodegenerative diseases like ALS and frontal temporal lobe dementia. where TDP43's misregulation is implicated. So, what are some ways we can tip the scales towards balancing the degrading effects of neurodegenerative diseases? While experiments provide valuable structural data, molecular modeling offers insights into dynamic behavior and energetics that are difficult to observe experimentally, leading to a more comprehensive understanding of complex biomolecules. Molecular modeling enables the simulation of biomolecules' motion and interaction over time, essential for comprehending diseases like FTLD, where abnormal dynamics and interactions play a role. By exploring intricate interconnections within biomolecular systems, such as protein protein and protein ligand interactions, molecular modeling reveals the underlying causes of diseases and potential therapeutic targets for FTLD. Additionally, molecular modeling contributes to the understanding disease mechanisms, including protein aggregation, structural changes, and interactions with cellular components, crucial for identifying targets and designing therapies to restore functional balances disrupted in diseases. In drug discovery, molecular modeling offers a flexible and adaptable platform for screening and designing potential drug candidates targeting specific biomolecular components involved in FTLD, accelerating the drug discovery process and increasing the chances of finding effective targets. So, I embarked on a preliminary investigation employing molecular dynamic simulations to explore the effects of mutations on IDR, on it, which is intrinsically disordered region, of TDP43. The underlying purpose was to attain profound wisdom concerning the intricate interplay of these mutations on the protein structure and behavior. By unraveling this delicate balance between function and disease within biological systems, we hope to unveil the essence of harmony that governs their existence. The yin yang symbol highlights the most constant aspects of being, the enduring truths. Some may question their reality, but they are indeed very real as they stand the test of time. In life, there are always things we can grasp and things we cannot, and they all contribute to our experience of being alive. Thank you. That means I did a good job.
Thank you so much. That was so exciting. So I know nothing about science.
Same here.
Get the philosophy and the metaphor of the yin yang. So I'm wondering, hearing your peer fellows speak about what you brought with philosophy to science. How do you see moving forward? What is the science bringing to your understanding of philosophy?
Well, in the reverse, I saw, based on my lab, this is my lab right here, y'all. These fine people right here. Can you believe it? Every last one of them showed up for me. So that speaks for itself. But anyway, I can use them as an example to answer your question. You know, each one of these people, Murat, Nate, Paul B, he's Paul number 2. Obviously, Jarrell Joseph, Nicole, Alina, and Ananya, they all specialize in something different. And then as they come together, they're not forming a whole of the whole, right? But they're part of the whole. So, you know, Murat, I kind of see as he's a cagal body, right? And Nate, he is a rigid body, right? And Jarrell is the nucleus. And Paul B, he's what should we make him, Nicole? He's a stressor, yeah. But anyway, you know, they all work together in unison, and their goal is the same. You know, they all, put their little parts into it, but their little parts add up to something that's going to do something good in the future. You know, they might not. None of us may live to see the results of this, right? But the interesting thing is that we're willing to. This is an example, a great example of, you know, putting your biases aside and what you think you know, and you put that aside to come together to formulate something to get to know, right? And that is the great thing right there. You know, the getting to know process is what it's all about, and that's what's special. And then not only is it special, it's a great thing because of the inclusivity part. You know, look at all this, look at the diversity in there. You know, there's a lot of diversity in there, and you need diversity in order to move these things forward. And that's why we're all sitting here right now, right? So there you go.